Hello, everyone. We are ready for the first lecture series on Philippine Planktology. Maayong hapon, magandang hapon sa lahat. Good afternoon and tajahao. Okay, so thank you so much again for joining us today with the last, but of course not the least, a speaker of the first lecture series of the Philippine Planktology. This is brought to you by different institutions from different locations around the world. We have people organizing this from UPV Miagao, and then of course Louis Lopez and me here in Taipei, Taiwan. This is made possible with our collaboration with the Institute of Marine Fisheries and Oceanology in CFOS UP Visayas and the Plankton Society of the Philippines. I'm gonna try to make a quick roll call of our participants today. We have about 250 registrants as of this morning, and a lot of them are from UPV, UP Diliman, MSU IIT, Xavier University, Partido State University, University of Southeastern Philippines, MSU Gensan, CIFDEC, Central Mindanao University, Q Quatics, Visayas State University, MSI UP Diliman, Ateneo de Naga, Bulacan State U, Cebu Technological University, Central Luzon State U, Philippine Science High School, Department of Education. We have from Pampanga State Agricultural University, Aklan State U, Angeles University Foundation, Palawan State U, CVSU NAIC, Isabela State U, De La Salle University, Bicol University, Our Lady of Fatima, Batangas State U, and Napocor. And of course, I think there are more people who just registered. So welcome everybody to the first lecture series on Philippine Planktology. Just a short recap of our house rules. We have, of course, we are asking everyone to please hold your questions until the end of the presentation. We will entertain the questions during the open forum. If you want to ask your questions, you can raise your hand and then wait for the moderator to call you in. And of course, you can also type in your questions in the Q&A box. Of course, we are asking, please do not spam our chat box. And kindly accomplish your evaluation form. This link will be posted later on the Philippine Planktology page so that you may claim your e-certificate. This is our short program. This is going to show, to tell you that we have at least 30 to 50 minutes for our speaker, and then we will have 20 minutes for the open forum. To open the program officially, we have the director of the Institute of Marine Fisheries and Oceanology to give us the opening remarks. Dr. Sheila Santander de Leon.
Good afternoon, everyone. Thank you, Christy. First of all, I would like to thank the Plankton Society of the Philippines and the Philippine Planktology for having INFO, the Institute of Marine Fisheries and Oceanology, as partner of this webinar series. The first two webinars were actually very informative and interactive. We received a lot of questions during the open forum. And I know that we have inspired a lot of students and budding researchers to follow the track of plankton research. And this is actually one of the goals of the webinar. Speaking of which, to those who are planning to enroll in graduate programs, our MS Fisheries major in Fisheries Biology and MS Ocean Science are now open for application for new students. Ma'am Christy is actually our faculty here, so if you want to take the plankton research, do join us. But of course, we have a diverse group of faculty that can guide you on your chosen research. So today's webinar is the third and the last of this webinar series, and we are privileged to have Sir Willie. Thank you very much for being here this afternoon. And but I hope that we continue these discussions in various other platforms to create collaborations and integrate our research efforts. Welcome everyone and have a pleasant afternoon. Thank you.
Thank you, Ma'am Sheila. Our speaker for today, we are honored to be joined by Dr. Wilfredo L. Campos. He received his PhD from University of Hamburg, Germany. He is currently a professor in the University of the Philippines, Visayas, and the chairperson of the Division of Biological Sciences in UPV. Dr. Campos is a marine biologist with interests in biological oceanography, coastal ecology, fisheries biology, marine protected areas, and fisheries management. He obtained his Doctor of Natural Sciences in Marine Biology from the University of Hamburg, and he has authored several publications in peer-reviewed journals, conducted over 50 researches in various aspects of marine biology and fisheries. He was also able to train a lot of students, and that includes me, and I'm very happy to have been trained by Sir Willie. And he currently heads the UPV Ocean Biolab. Thank you so much for joining us, Sir Willie. Now, please take it away, sir.
Okay, thank you very much, Christy. Is my audio okay? Or should
Yes,
I?
po.
Okay. So yeah, let me just share. Okay, good afternoon, everyone, and thank you for attending this webinar. Um, I will talk on using fish egg and larval distributions in designing ecological networks of marine protected areas. Um, this, I'll be presenting some of our work. It's not just truly our work. It's really a lot of collaborative work, especially with our partners, the physical oceanographers from the lab of Dr. Villanoy. So really the authorship here is really more plural than just myself. No? Um, anyway, ecological connectivity is really a fundamental, a set of fundamental ecological processes. No? It concerns the movement between populations, movement of individuals in populations, or their gametes or propagules like larvae, the movement of genes, or even non-living material between populations, communities, spatially fragmented habitats and ecosystems. It's believed that ecological connectivity is an essential part of designing marine protected areas. Um, Ecological networks of marine protected areas are really systems of major or core habitats connected by ecological corridors, and they're established um, in systems and ecosystems that have been fragmented for whatever purpose to conserve biological diversity. And here in the country, it's really to the hope of restoring the productive potential of these systems. So I'd like, in this context, I'd like to share with you some of the results of two of our studies, um, looking at fish eggs and larvae, abundance, their composition and distribution, in the context of marine protected areas. Um, so the first study that I'd like to share with you is our study on the east coast of Mindanao. And here I'm showing um, the bifurcation of the north equatorial current. Um, and this is the eastern coast of Mindanao. South of the bifurcation, um, there's the habitats are really open and exposed along the coast. They're because of this, they are probably less productive in some ways. No? Um, and along the Mindanao coast, the eastern coast of Mindanao, the as you can see, it's aside from Leyte Gulf, the shelf area is really very limited along most of Mindanao, with just several embayments south of Leyte Gulf. And because of this, um, the source of recruits, for the different habitats, including reef areas, along the eastern coast uh, is really limited, compared to other areas within the inter-island regions. And one very strong characteristic of this area is the strong southward flow along the coast. Okay, that's Leyte Gulf, and then these are the embayments along the east coast of Mindanao. So we did a series of plankton surveys dating back 2003 to the most recent one was 2017. So I'm trying to put together these studies to to um, to give us a picture, no, of a larger scale picture of what might be happening. No? And these studies had in common the goal of determining the composition, abundance, and distribution of ichthyoplankton, no, eggs and larvae. And they used comparable methods, no, using surface horizontal tows of 10 to 15 minutes, using the same kind of net, no, and um, IDing the larvae to family level. Well, first survey was done in June 2003, covering Surigao Strait here, what I call the inner Leyte Gulf and then the outer Leyte Gulf. Not just the Gulf, but actually the shelf area of Leyte Gulf. And then a second set of surveys, not soon after, no, in Lanusa Bay, covering 30 stations, and in Lianga Bay, covering 34 stations. These were done in the year after. No? And the third study was also done in the same time of the year, late summer, done in Hinatuan Pass in 2007. So um, there were largely different groups of people, but again, um, belonging to the same lab and using the same protocol. Together with this was studies on the hydrography that were done by the physical oceanography group led by Dr. Cesar Villanoy of UPMSI. They did the studies in August 2005, including measurements of vertical profiles of temperature, salinity, and chlorophyll in this area. No, uh, this is still part of Surigao del Norte. Well, this part here is Surigao del Norte, and mostly Surigao del Sur. No, this part, and they measured current speed and direction with the use of a drog, no, and wind speed and direction. And from these empirical measurements, they used them to in a modeling effort to simulate the dispersal of larvae, which we'll look at later. No? Aside from this, we did a small drift card experiment. Actually, it wasn't really a full-blown study, wherein we used about 100 drift cards here shown here. They're really postcards which were enclosed in a piece of plastic with a strip of styrofoam so it would float on the surface, and a weight which we used an ordinary nail. So we actually back then in 2003 we contributed to garbage in the ocean actually, but the results were actually very helpful. So we released them in three sites, and we got these were all self-addressed and and stamped cards. So we were able to get return postcards within a period of 18 days. So in terms of fish larval composition, the red areas here are the stations where which we refer to as the inner Leyte Gulf. No, the composition of larvae here are mostly reef associated species, but equal portions of demersal, soft bottom, shallow water, and pelagic species as well. No? And then a few deep water species. In Surigao Strait, no, there seems to be a concentration of larvae in Surigao Strait, no, as the water is channeled through the strait coming from Leyte Gulf. No? So we have more than half of the larvae that we've seen here are reef associated, and the rest are really shallow water pelagic and demersal species. No? In the outer shelf of Leyte Gulf, we see that reef-associated larvae still dominate the catches. No? And the smaller surveys in Hinatuan Pass, um, this is really in waters between the many islands here, no? We still find a lot of reef associated species, and then more small pelagics. And in both Lanusa Bay and Yanga Bay, similarly dominated by reef associated species. No? So in terms of densities, In the inner Leyte Gulf, we found densities that were larval densities that were quite high, no? Okay. Um, and Hinatuan Pass, smaller densities here, no? Okay. And in Lanusa Bay, also lower densities than what we've seen in Leyte Gulf and even in Surigao Strait. No? So if we just summarize and put this broad information together, there is about 3 to 6 times more reef-associated larvae or larvae of reef-associated fish in this portion of Leyte Gulf, which is inner Leyte Gulf and Surigao Strait, 3 to 6 times more than in the areas south of it, especially in the embayments. No? Now, for the larval dispersal simulation, um, uh, the simulation you make use of 3 source sites. No? Here in the southern tip of, I think this is Siargao Island. This is part of the Hinatuan Passage here. No? And then in south of General Island in Surigao del Sur, this is Lanusa Bay. No? So 3 source sites, and the model follows or traces particles located each day for a 30-day period. No? So the tracks here, color-coded tracks, refer to the respective sites, source sites, no, and the path shows essentially for the island of Siargao in 30 days. Well, the pathway is towards the coast and not really hugging the coast, but towards the south and actually into and outside of Lanusa Bay. And it's the same thing for sites that are close to the mainland um, that have to go through the small channels between the islands. They remain close to the coast and hug the coast in Lanusa Bay. No? The same thing for here for General Island. No? So the general dispersal is southwards and along and close to the coast, actually. No? So the results of our drift card experiment showed that these are 2 of the release sites. We released here in San Pedro Bay, actually, and the numbers, encircled numbers, are the number of days from the release date that the respondent, they were all fishermen, noted down on the card where when they retrieved it and where they approximately where they retrieved it. No? So those released in inner Leyte Gulf stay within Leyte Gulf, but a portion of them are transported eastwards towards the edge of Leyte Gulf. No? Those in Surigao Strait, those that were at least returned to us, all of them were um, were drifted towards their residences in the islands close to Dinagat. No? So there is an eastward transport from that site in Surigao Strait. No? Although we know that from many oceanographic studies and many other um, investigations that there's a very strong uh, component of flow from the Surigao Strait into Bohol Sea. Okay, the third site was in the outer edge, just south of Giwan, no, where the flow is predominantly going south, and showing that within a week, um, the drift cards are are well reach Katil Bay, and well in Mati the 18 days, well for sure within 3 weeks, ano maabot na sa Mati, no? Okay. Okay, that's Yang Gabi. Okay. So from this, no, there are many more things that we did in the study, but I'll just um, give you the broad conclusions from these results. No? So from in the inner Leyte Gulf, we've shown that the reef larval concentrations are greater than 3 times more than what you find south of Leyte Gulf. No? Okay, and then the movement of water dispersing the larvae is towards the east and then south along the coast of eastern Mindanao. No? But a portion of this is also moved or dispersed towards the south into Surigao Strait, where A portion of that is transported into Bohol Sea, but also a portion of that is moved eastwards towards the inter-island areas and passages in Inatuan Pass, and then further south, very close along the shore, southwards. Okay. And on the right here is I'm showing actual observed current flow in direction from an experiment done in 2007, the Felix 2 experiment, showing the vectors showing direction and magnitude of flow. So there is really a very strong flow from Surigao straight into Bohol Sea, bringing in larvae actually from Leyte Gulf. Okay. So this dispersal covers almost the entire coast of eastern Mindanao within a 2 to 3 week period, which is a typical larval duration in the tropics. And what we can infer from this is that Leyte Gulf is a substantial source of fish larvae, not only for the eastern coast of Mindanao, but also for the areas along Bohol Sea, north and south portion of Bohol Sea. Okay, and a second study that I wanted to share with you is on a much smaller scale. Study that was done in Danahon Bank in northern Bohol. No, the Danahon Bank is a double barrier reef system. It's a unique geological feature. There are only one of 6 known double barrier reefs in the world. Danahon Bank is smaller than the others. No, it's the only one in Southeast Asia and covers over 270 square kilometers. No? Okay. That's at the Nahom Bank area. This area has been heavily exploited for several decades, and up to this very day, fisher density remains high, and even unsustainable and destructive fishing activities remain and are still being practiced in the area. And these have contributed a lot to extensive depletion of resources in the Nahon Bank. No? So compared to many other areas in the country. No? Okay. So the study that we conducted also wished to aim to determine egg and larval distribution patterns to provide insights on sources and potential sinks, origins and areas of potential settlement of recruits. Um, um, in the hope that this strategic approach um, um, contributes to the functional and ecologically functional MPA network by ensuring continued connectivity by protecting those areas and allowing them to recover their productive potential, um, enhancing effectiveness of protection, and to hasten the recovery from disturbances or resilience. Okay, the survey was done in December 2005. Phytoplankton, zooplankton, ectoplankton were collected, pretty much the same manner that was done in Surigao. There were 24 stations located here, shown in blue, covering the eastern portion of the bank, the central portion of the bank, and at least a portion of the western portion of the bank. Okay, surface tows were done during the daytime with a plankton net tied to the outriggers of the boat. Again, we partnered with the hydrography, the physical oceanography group of Dr. Villanueva from UPMSI to do the hydrography, which they did also in the same month, using drugs again to measure current flow and direction with measurements of temperature, salinity, chlorophyll A, looking at 23 stations at different stages, the ebb and flood stages of the tides, and employing simulation modeling to look at dispersal. So I'll just summarize here the gross results. This shows the fish egg densities. Fish egg densities um, are actually low compared to what you typically find in other reef areas in the country and in most other coastal areas that we've studied. Egg densities on the average are 4 to 10 times lower than we've encountered in other areas. Although there are some areas where egg densities are concentrated, on the whole, eggs are mostly dispersed. Okay. for a semi-enclosed or for a contained system like the Nahon Bank, no, these are the banks that are submerged, no, and they're really extensive, no. So it encloses or contains the shallow water portion into what we call the Nahon Bank system. No? So for such a contained system, these egg densities are rather low. So fish larval densities are typical and on the higher upper range of typical coastal densities. They range from 0 to over 1,000 individuals per 100 cubic meters. No? So uh, reef fish biomass in all over the Nahon Bank is really low compared to many other areas in central Visayas. No? But again, what stands out here in this study is very high larval densities, no? In spite of low overall fish biomass. No? So we see that the larger markers here denote higher densities. No? Okay, and we see that there are higher densities on the margins, no, of the central, on the periphery of the central bank region, where you have the islands and the most extensive submerged bank areas, no, of the Nahon Bank system. No? So that we believe that the low concentrations of larvae here are um, are probably linked to settlement of larvae in this area. No? Okay, so that we believe that this area of central Danahon Bank serves as both a sink of larvae coming from the eastern portion, you know, and as a source also of larvae moving into the western portion. No? Okay, but we'll talk a little bit more about that when we look at the hydrography. But the fish larval composition, the top 10 families contributed more than 80% of the fish assemblage, and they were not dominated by reef fish. No? Okay, they were dominated by soft-bottomed demersal species. No? But again, this might be a function also of the time of the year that the survey was done, which is December. No? So these are typical, the dominant families in the catches, no? Therapontids and Leognathids and uh, I think these are dragonets, no? And then we have epipelagics, we have carangids, no? And we do have clupids as well as and growlids, no? And then we have your typical reef associated species, no? Dominated by gobids, apogonids, and then of course your pomacentrids, no? And then your even deep water species, your myctophids, and then your bregmacerotids, no? Okay, the general flow of water um, from the drug experiments shows a predominant movement of water from the east going to the west, no? um, during both seasons. No? During the northeast and they shift only with seasons in the eastern portion of the bank, wherein there is substantial inflow from Surigao Strait, no, into the Nahon Bank during the northeast monsoon, and during the southwest monsoon, it's coming from the north, no, from Camotes Sea, and there's net movement at least from the eastern bank into Bohol Sea area. No? Okay, but the simulation experiments, larval transport experiments or simulations, no, again followed 30 days. And the boxes here, the color-coded boxes, correspond to the sources at which the simulation models used and allowed particles to be traced for 30 days. No? So the colored-coded small particles represent the path, no? That the virtual particles, virtual larvae, if you want, would take according to the model. No? So that in the um, eastern bank, outer eastern bank, this is the inner eastern bank. No, most of the particles here are not really dispersed very far, but are retained within this area. No, okay, okay, we're retained within that area. In the inner, if there are sources in the inner um, uh, east bank, no, we can see that the color-coded paths are towards this region here. No, okay, so there is dispersal towards the central bank. No? And if the sources are in the central bank, you know, and we see that the movement, the dispersal is towards the western part of the Nahon Bank. This is during the northeast monsoon. During the southwest monsoon, regardless of where the sources are, all of them show predominant dispersal towards the west. Okay, so that as mentioned earlier, I'm sorry, um, the west portion of the bank receives larvae from sources further east. So that in the central bank, no, okay, this area is the central bank, okay, central bank portion, no, serves as a sink for propagules coming from the eastern portion of the bank, no, and also as a source of larvae from the central portion of the bank going to the western portion of the bank. So the western portion of the bank really seems to function as a sink of recruits or propagules coming from sources further east. Um, with that information, uh, we use this as input into designing a network of MPAs in western Danahon Bank. This is where the white square is shown. No? It covers 21 MPAs in 5 municipalities, including a portion of central Getafe in the central portion of the bank, all the way down to Tubigon. No? Okay. Um, and although this network is a management network, no, uh, because uh, in design and in determining which municipalities to include, um, the results of these surveys were used, no, as part of the basis. No, we believe that it's a management network of ecologically connected MPAs. So the Nahon Bank is ecological connectivity in the area is physically contained because of the physical and topography, physical nature, structure, and topography of the system. No? And in far as governance is concerned, all of this is within a single province, no? A single governance organization within that level at least. So the networking of management is easier, more readily done. No? And therefore the higher there's because of there is a higher prospect for success. But of course, success is to be determined after. No? Okay, the network was established in 2016, and I am not really very familiar if what efforts have been undertaken to maintain this. No, but the challenge, I guess, no, in many parts of the country, is how we can adopt a similar system, no, that we have seen here in the Nahon Bank, no, with more complex governance structures, no, where the connectivity transcends geopolitical boundaries, so to speak. So this is really a challenge also facing fisheries resources, no? Where very recently fisheries management areas have been established um, as a paradigm shift, if you want, into looking at how we should be managing our resources. No? Okay. So just to wrap up, no, our studies on ichthyoplankton abundance, distribution, um, very are because they were really tightly, if you want, tightly coupled with hydrographic study. They allow inferring ecological connectivity, in those areas, but not truly determining if ecological connectivity is really what is happening. Okay. And the question is, no, under the setup here in the Philippines, so is this sufficient? No, inferring ecological connectivity from these studies like this, no, is this sufficient to guide the design of ecological networks in the MPAs? Um, we've tried also including the distribution of larval developmental stages. I mean, this actually provides more information, but you also see a lot of natural variability, so that the really gives no clear patterns, but it does, it does provide more information. So um, um, we know that larval dispersal is a major determinant of recruitment, or replenishment of populations. And but it's not the only major influence on that. There are many other variables, including the downstream habitat conditions in which larvae are supposed to settle, or early stages are supposed to settle and then grow into juveniles in mature stages. Um, the scales of ecological connectivity, which differ not only in space, but their scales also differ in space and time. No? Not only between seasons, but also between years. No? And also retention versus dispersal processes is still uh, really no fixed generalizations about this. No? And of course, climate change, what effects will these have on the hydrological and ecological processes that also figure into larval dispersal. So there are many things that we need to understand really to really include this ideal, this improve our understanding so we can incorporate this strategy into designing networks of ecological networks of MPA. So more work is needed, especially in the tropics, because most of the information that we know right now is coming from Australia or from California. And in the tropics, where spawning and recruitment is year round. Uh, but in spite of that, we do have, for example, in the Philippines, seasonal shifts in the changes in current flow, which will also translate into different, larval dispersal paths, if you want, no? Or at least magnitudes of larval dispersal, no? Including the archipelagic setting in the Philippines is also more complex, no? So there's a lot of information that we would like to have, no, for this to become more useful. No? And a major concern, of course, is the costly, time-consuming, and tedious work, involved in working with larvae. As we all know, larvae are in their early developmental stages. The identification alone is already a lot of work. Um, and we need to weigh that, the cost of all of this tedious work with the strategic gains, or possible strategic gains in enhancing the success or the impact of establishing many small MPAs in each municipality, or rather strategic gains in enhancing the success and impact of establishing networks that are ecologically functional, as opposed to establishing many small MPAs in each municipality. No? Okay, it's not only a topic of academic interest, but it also has very practical interests, because it enhances knowledge of these larval dispersal paths, and ecological connectivity, enhances the recovery of productivity, and even of these MPAs, even with the distant goal of protecting 10% of coastal waters, which is their overall recommendation, uh, for conserving biodiversity and production recovery. So I'd like to suggest ways forward. Definitely, there is a need for a combination of approaches. No, genetics or parentage analysis can provide a finer resolution to addressing this problem, or this addressing ecological connectivity. And from the studies that we've done, for example, the dispersal paths that we've seen to identify can limit or identify the geographical coverage for such genetic studies. Definitely, hydrography and simulation studies are necessary, and this is really a gap or a limitation, a serious limitation in the country. We need to develop local expertise in these aspects of oceanography, and even in early stage biology, not only of fish, but also of invertebrates as well. No? Okay. Of course, ecological connectivity does not only concern early stages, it concerns also movement of your older stages. No? So that's another aspect of connectivity that that should be considered. And of course, the scales in time and how this varies in time. So we need to develop a monitoring system of connectivity, monitoring connectivity over time. And the more practical considerations are we need to effectively communicate the benefits of uh, using ecological connectivity to policymakers because they're the ones who make the policies, they are the ones who make the funds available for this research. No? Again, it's not just an academic exercise, but it's also of practical interest. No? And of course, the issue of governance structure that allows the management of resources with fluid borders. No? Okay, again, the recent adoption and establishment of fisheries management areas in the country is a step in the right direction and also in this direction. No? So possibly it's also ripe for studies like this to begin again in on expanded coverage. Uh, I hope. No? Okay. So allow me just to a couple of minutes to acknowledge because this is the only one of the rare opportunities that I can acknowledge the work of many project staff and assistants in the lab. No? So these include DM Estramadura, Nonoy Bildia, August Santillan, Noel Evano. We have Mary Noblesada, of course, Christy Acabado is always here. And more lately, we have Luke, Felix, and Dexter Piloton, and many others. No, I cannot including me. It's going to be a long list. No, many others from then and now. Of course, we've worked very well with the group of Dr. Cesar Villanueva, the Visual Oceanography Group of MSI, and really, we need more groups, such groups, so we can work together in different regions of the country. And we need to develop the capacity in different regions of the country. You know, if we decide that this is the path, the way to go. No? And also, very grateful to for many of the discussions for many years with many colleagues on this. No? The studies that we've been done in this area of interest were funded mostly by DOST before Picamard, now by CARD, no? By the Fish and Ecofish projects, and by Conservation International. So with that, thank you very much for listening.
Thank you very much, Sir Willie, for that very informative lecture. Those studies go back to what 2007, right, sir?
Yes.
That's
2003, I'm
very
sorry, 2003.
2003. That is very interesting. Also, I would like to say that this webinar is a very good venue for different planktologists around the country to meet and to meet everybody. I'd also like to say that this is a good reunion for your previous RAs. So really, DM Estremadura and August Santillan are here today with us.
That's good.
Yes. I think we can proceed to our question and answer. Sorry. Okay. So we have a few questions from the audience. First, the question is, like for example, in your study in the Nahon Bank, how do you usually determine that an area is a source of fish larvae?
Well, we inferred it. No, so it's very difficult to really determine and identify its source. But looking at the distribution of early stage larvae and up to later stage larvae, their abundance and how these abundances are distributed in the bank area. Uh, the inference is because uh, there were very few uh, larvae, especially of the later stages in the central portion of the bank. No? Um, it's unlikely that they were transported more to the outside of the Nahon Bank than they were inside further west. No? So our interpretation of that is likely they settled in the central bank. No? But these are from larvae coming from propagules or that produced in the eastern portion of the bay. No? We did not really study spawning and really of fish or other organisms in the eastern portion of the bay or any portion of the Nahon Bank. No? Well, we just um, um, used larvae and their also genetic distributions to infer this, together, of course, with the hydrographic studies. No? So, yeah, to answer your question, that is what we did, no? And we did not make any studies that will directly allow us to determine that spawning occurred here, that we could follow and really trace that the larvae that settled here came from this location. Uh, no, we were not. We did not look into that.
Okay po, sir. Maraming salamat. We have a question from the audience. The drift card is a very interesting method for studying dispersal patterns. Any suggestions to increase percentage retrieval or alternative methods for more data generation?
Um, well, I guess now we can use no, now we can use more biodegradable materials. No, um, at the time we were still uh, we were still not using cell phones. No? So hindi pa uso yung magpapasa ka ng load or anything like that. So we had actually thought of how we could reward or send rewards to people who would send back the postcards. No, and we it was not very easy to do that. No, but I guess in activities like this, no, including in tagging experiments, for example, no, short-term tagging at least, no, they do give T-shirts, for example. It's easy to enough to pass a load, for example, no, give some incentives, no. But again, in addition to that, I guess dissemination would be much more important, no. And really, we didn't have the opportunity to disseminate this. Aside from really just explaining in that small card what the importance of returning the card would be to uh, to research, no? And knowledge, no? Yun lang. So I guess there are ways now to improve that compliance, no, or if you want retrievals, no, or and those would be some of them.
Okay po, sir. A question from an attendee. How does the community in your study sites respond to these efforts by the academe or NGAs by performing such studies? Did they show willingness to participate in managing these resources and undergo technical trainings?
Yes, when we did our work we were involved in the establishment of the MPA network in Danahon Bank, no, and this was done in actually what 12 years after, 11 years after the study was done on the ectoplankton, no, and we used this as part of introducing the project, no, of establishing a network of MPAs, no, and that was very interesting to them, and there's a lot of questions from them, but it was received uh, um, with a lot of interest, and I think it also provided motivation for them to go ahead with the management, because eventually the connectivity idea settled in them, and they I think this was important to them. So I think it was they took it well, and they was also motivated the entire effort.
So we can say that the LGUs also show support in terms of like financial, or they also express interest in the trainings of the community.
Yes, we did not really train them in doing the ecological surveys, but what we did was we conducted capacity building for management, no, capacity building for doing their own habitat uh, assessments, no, of shallow coastal water habitats, and workshops for how to put together a management plan. Yes, the LGUs, all the LGUs involved were very very cooperative, and they were interested in and they had their counterparts, no, for supporting the effort.
Sir, madami po tayong questions.
Go ahead. Okay lang, okay lang.
We have enough time, I guess. So someone is asking if is there any research group in the Philippines that implement the use of DNA metabar coding to assess species composition of fish larvae and eggs for monitoring?
I'm not aware of any, but I would be very interested in knowing how it goes. No? I have many questions about this simply because I am not familiar with the methods. No, but I maybe there are for fish larvae. No, I'm sure for fish there are, but for maybe they are for fish larvae. And yes, I'm just not familiar with any particular group.
Okay, sir. A question from your wife. So, what do we know about larval transport or dispersal in the Visayan Sea, and how is it useful, the knowledge on this, given the nightmare in governance that the Visayan Sea has?
Oh, well, first, the nightmare of governance in the Visayan Sea is not only in the Visayan Sea, it's going to be an issue that we need to, it's a challenge, no? Issue that we need to tackle in all the fisheries management areas, no? Which transcends administrative regions now. No? Anyway, the Visayan Sea, I wish I could answer this question. No? And I think the very early stages of our work here, no? And even DM can attest to this. No? We've tried tackling this question and really now, for example, we're seeing for even stocks of sardines, and these are already mature sardines. No, we see differences in the composition and of the stocks. No, sardines in Acid Gulf, which is the northern part of the Visayan Sea, and the southern portion of the Visayan Sea. No, and there's really no physical barrier. No, and although there is a somewhat, I wouldn't call it deep, but a somewhat deeper canal that runs from east to west. No, that essentially, if you want, right down the middle, if you wish, so you can have a north and southern portion of the Visayan Sea. It's not very deep. No, maximum probably of 100 meters, but it's probably between 50 and 70 meters, whereas the rest of the Visayan Sea is probably of the average of 20 meters. So it's not a very, very big difference. No? Um, really, that's where we really need our oceanographers, to look at maybe on a finer scale than what we see as the broad descriptions of water movement, during the seasons. No? Perhaps because it's a very shallow area, there are many islands also involved, but also on the bottom there are also a lot of shallow areas. No, that probably also um, has a bearing on the flow of water, how this changes with season. So really, it is still a question to us. No, we do have larval information from this. No, but probably not at the scale that we need to be able to address this question. So I don't think I can answer that.
Okay po, sir. Actually, I have a question too. You mentioned that the Nahon Bank has lower densities than typical coastal regions. So you also said that this is possibly because it is the source and the sink of larvae at the same time. So I was thinking if we have hydrographic information on the turbulence that is created by the shallow banks in this area, which could also possibly kind of remove the plankton away from the area.
That's possible, especially in the central area, no central bank area. No, um, yes, that's where the issue of scales, no, in space come in. No, um, yes, those are like really fine questions that we really cannot answer on that scale. No? So maybe if there were other opportunities in the future, maybe that can be addressed. No? One thing that we've also noticed is when we look at the distribution of crab megalopa, although we weren't able to identify if they are plutonic crabs or not. No? Um, we see the concentrations in the outer banks, no? In those extensive grass beds that are submerged. No? We see them there. No? So we don't know if it is really an active movement to spawn there, or maybe if it's a physical uh, process that concentrates them there. No, we did not go into that resolution, no? But those are
Uh-huh.
questions that are interesting to look into.
Okay po. Kaya po, may isang question dito. How do we address the lack of physical oceanographers in the country? Do you
I think
have,
also,
yeah.
I think I think Dr. Villano is here, no? But
Yeah, he is.
how do we address that? We need more people to take up physical oceanography, and we need more to hire faculty in different regions and not just in Manila, no? So that those that are in different regions might find it more easy, no, and more less costly for them to study in Manila, no? But again, now if remote classes continue, I, I mean it opens up so many possibilities for this. No?
Okay po. Another question, madami po tayong time, sir.
Yes, yes.
So, before we can design an ideal MPA connectivity, how many times do we need to conduct the survey since underwater current changes depending on the period of the year?
That's true. That's very true. No, and in shallow waters, no, we haven't really even looked at larval behavior, no, with how it differs from day and night, how it differs with the tides, etc. No, but in my own work in the North Sea, no, which is 40 meters, it's very shallow, no, 40 meters. The vertical distribution of larvae was very, very noticeable, no, and there's a pattern to it which I try to explain. So even in shallow waters, even within the Nahon Bank, you can find that. In many shallow inter-island waters in the country, that's the same too. No? So even the vertical distribution is which is very important in determining the location of the larvae because, like the question posed, no? Vertical currents change in any given time. They're different, no, in the water column, and also they change with the tide and they change with different times of the year as well. No, so these are all, I guess, part of the question of scales. No, but I guess what I tried to show in the presentation was, in spite of the lack of many of these details, no, um, there is already a great deal of wouldn't say information, but um, it's really a very big eye opener to say at the very least, no, to to see how the hydrography woes and where the larvae are with respect to this hydrography, and they in many ways are consistent with one another. No, it does not prove that there is connectivity between these areas. No, but it allows you very to infer that. No, and it's very difficult. You will need to do very fine scale and directed studies to actually disprove that. No? Okay. Um, so I'm not saying that studies like this are the end of it. They certainly are not. No, but they certainly are not useless. No, I think there's a lot of meaning to them. No, and we can dissect and do more studies on a finer scale um, to get more information about to answer those more specific questions. No?
Okay po. Maybe last three questions. Uh, okay, so in the future po, can you think of other parts of the country that you may probably want to work with that possibly needs the same kind of information on larval dispersal to create MPAs and MPA networks?
Um, I think there are many. No? There are many
Yeah.
areas, so many areas, and I think I guess the challenge is in those areas where you have inter-island areas in general. No, it's the flow of water will depend on many things, no? And you can expect what the water flow, movement of the water to vary on different scales and in both space and time as well. So the work there is going to be more challenging. No, um, in areas that are more open, where the flow of water is less obstructed, then maybe it will be less complicated, but I wouldn't say easier to infer. No, but there are many areas where we can do this, and I would think that whether these areas uh, are open areas like the margins of the Sulu Sea, for example, no? Or inter-island, like in the Central Visayas area, or in the Colyon Island group, or Palawan area. All of these areas are going to be interesting. No? I just want, because of personal, personal, if you want, personal, not preference, but just a personal interest. No, I'm interested in what is happening in the Colyon area, because there's so many islands there, so many islands. The bottom is so heterogeneous. And from our studies in Sulu Sea, which I wanted to include also, but it's a much larger study, um, it looks like it's a major source of larvae for the central and southern portions of Sulu Sea, at least for certain kinds of larvae, for reef-associated larvae. So that would be an area of interest for me and personally. But again, many of the areas are going to be uh, um, interesting, equally interesting. As far as priorities, I guess it will all depend on the resources which people depend on in those areas. No?
Okay po. Do you think the research in the Nahon Bank is the best model for ichthyoplankton MPA research? Have you done similar small-scale research in other parts of the country?
Um, we've done several studies on ichthyoplankton that are of a smaller scale, than the Nahon Bank. What is, I guess, most meaningful in the Nahon Bank is because the system, I call it a contained system. Of course, it's open, no, but for the most part, the submerged banks themselves, physically contain uh, the connectivity. No, it's not, of course, 100%. No, but if you compare that with more open areas, like say Visayan Sea, for example, no, or the Sibuyan Sea, or even Ticao Pass, Samar Sea, those areas, no, or even the embayments along Bohol Bay, no, Bohol Sea. They're much more open, less contained, no. So I wouldn't say it's the best, no, but I think the results were more meaningful, not because it was intended that way, no, but because the opportunity was there to do it, no. Uh, and Yes, so I guess other places are not going to be as contained. So in many of our other smaller surveys, um, the areas were not contained, so that the scale at which we did the study is not as meaningful as what we um, were able to, as the amount of meaning that we were able to glean from our study in Bohol. No? So really, how to answer that question? Really, you need to basically look at the map, study the map, look at the bathymetry. You have to talk to an oceanographer, somebody who knows hydrography, um, because really they will have a very, very good handle on what the physical processes are, where they're going to be more meaningful, no? And at what scale they will be more meaningful. No? So that's why um, you really have to work with oceanographers, and I think it's very, very important that um, we work together with them. Yeah.
Okay po. Yun po, opportunity pag nandun, they gawin, di ba?
Yes, yes, yes.
Yes. Sige po. What is your stand on House Bill 7853, especially that it poses severe threat to municipal waters? Will these also leave a huge impact to the population of fish larvae?
785 is a proposed bill to to allow larger vessels, commercial vessels, to fish closer to the shore, di ba?
Yeah,
No,
I
I think
guess
that's it.
so.
No, yes. Well, your larvae are going to come from your spawning, your mature, no, mature portion of your stock, no, and I think there's little question about for most stocks being more abundant proportionally closer to the shore. No? So that if you allow larger volumes, no, of to be caught of your potential spawners, no, in coastal closer to the shore, then you're directly affecting the reproductive capacity of that stock. No? Okay, but let me just go further than that. No? Um, because 7853 is no no is a matter of both municipal fishing areas and the commercial fishery, you know. And that bill itself will have its own argument, no? And really um, it's a much larger argument in terms of livelihoods, no, for small versus larger operators, no? But as far as the biology is concerned, no, the stocks are concerned, no, what's important to them is, of course, the habitat, no, and essentially how much of them are being taken away, how much of the spawners are being taken away. And given that the habitats are not going to be destroyed by any sector of the fisheries, no, uh, and it's just the amount that will differ between the sectors, no, it really doesn't matter to the stock who is catching them. No? What matters to them is how much is being taken away. No? So whether it's being taken away by a single fisherman, by single fisherman operations, a billion of them, or a handful of commercial fishers, the total amount that's being caught is no, no, yun ang suma total for the stocks. No? Okay, given that everything else is equal. No, okay, of course that doesn't mean that I'm supporting 7853. I'm just, it's another dimension, and to me, it's the biological dimension is at the root of everything. No? If you don't have your stock, the capacity of your stock to produce, if you impair that even more, then I think it's elementary. No, it will not affect. It will affect your egg production, your larval production, and the recruitment the next year. No, that's clear. No, so yes, but again, the issue of 7853 is again much larger than ecological connectivity. No?
Okay. Thank you po. Sir, last two na lang talaga. Hanggang
Okay
3:20
lang, okay lang.
naman po yung Q&A natin.
Okay lang, okay lang.
Dami kasing interesting questions. Sir, really, you often mention physical oceanographer. How about chemical oceanography? Does it have a role in fish larval distribution?
I'm sorry. I'm sorry. No, I keep lumping. I don't do that intentionally, no. But really, that's why I use hydrography, because really the application that what we was really the movement of water. No, but really there more. There's more to the physical aspects of the processes, no, in the oceans. So it includes, of course, chemistry and geology, as well as other aspects of biology as well. No? Yes, definitely chemistry. Affects that. No? But of course, the chemicals, the chemistry of your waters, that of course will include your salinity, your temperature, and the physical-chemical properties, which in a way will also determine your currents, your current systems in a gross way. No, okay. But I guess on the scales that we've seen, if we're interested in the no, uh, well, whatever the scale is, yes, chemical oceanography is also important in terms of nutrients, in terms of if you want pollutants as well, no, in terms of I mentioned earlier behavior of your fish larvae, and I believe chemical cues are important, no, very important, no, especially when they're far away. They're much work in the 1990s, much work was focused on visual perception of larvae and their orientation, no, as far as movement up and down and the resulting dispersal. No, I've always believed that olfactory cues are important, no, especially if you're far away from the coast, no, and mechanical cues as well. No? So um, we're not even beginning to look into that. No, it's abstract for most of us to look into that. For now, it may sound like a very academic exercise, no, but yes, definitely chemistry is very important as well.
Okay. What strategies can be employed by researchers to translate these types of studies into citizen science for a better appreciation of the study for coastal resource management?
Okay. For studies like this, no, for ecological connectivity, specifically for this kind of work, like I mentioned, we need to communicate this more effectively, no, and the venues for doing that are varied. We need to really take advantage of this. When I say we, the scientists, no, have to really take advantage of these opportunities, no, at both the local and maybe the regional scale, no, and for other scientific, no, scientific endeavors, um, uh, it's also well, of course, you have to get funding, no, and funding from agencies. It depends on their agenda, of course, no, and um, maybe this is not really a recommendation, but maybe a suggestion, really, and maybe just words of encouragement that I guess in any academic interest, be it purely academic, really basic academic interest, no, you will always find a way, if you think hard enough, be imaginative enough, find a way to get into the agenda of the finding agencies to insert your special interest. No? And it may be a little hard to imagine at the beginning, challenging to imagine that in the beginning, but there are no rules in this. No? So I would encourage the young ones to not to give up on your basic interests, but instead of doing that, you look at the agenda of your funding agencies and see where you can fit in. And I always say, itago mo, no? Okay, and then look for opportunities within that to look at your academic interests. No, oftentimes those interests that now might be purely academic, might not be once you show your results and you study them and get an opportunity to study them and show your results. No? And this is what we felt, no, in the beginning of our studies ourselves. No? We weren't sure that this was going to appeal to the OST, no? And so we hid it somehow, no? And we hid it well. Anyway, that's maybe just just word of advice, maybe. Yeah.
Okay. Okay, last 8 minutes. So, how do you think this kind of analysis be of use for science-based management of the fisheries management areas or FMAs in the country?
Uh, yes, I think like for example, the simulation part of it is, I think, very very important. No? But of course, you need data to validate, if you wish, what your models are showing, no, empirical information, no. But it's not only distribution and abundance that you can gain from this, no. Um, uh, Certainly, you need some modeling in some surveys like this to look into the productivity of an area, no? The seasonality of productivity, what contributes to the productivity, and how if there are uh, spatial differences in areas of high production and low production in an FMA, for example. Uh, is that contained the entire year in this area, or is that also dispersed somehow? No, and this eventually will this is eventually the path or the direction that we will move into. For a long time now, scientists have been really strongly suggesting that the management of our resources not be based on geopolitical boundaries, but based on ecological boundaries. No? And it's only recently that this has become law, adapted into law, and it's not perfect, of course, but it's a step in the right direction. No? So yes, there are many applications, and not just to your larvae and recruitment, but also explains the productivity, no, and of of an fisheries management area, and in part will also explain how the fishery operates, no? If they move within that fishery management area or not, or if they stop fishing at certain times of the year, what is they don't shift to other resources. Many things will depend on the productivity of the area. So studies like this may not be appreciated primarily because of the cost of the work, no, right now, but we are in that direction, no? So I think it's really time to really start building the capacity to do these kinds of work. So we work together, no, with the evolution of our or our governance structures.
Okay, last na lang po, sir. Have you encountered microplastics in one of your samples? What do you think might be the impact of the presence of these pollutants to larval dispersal?
Well, we have not really looked into microplastics. No, well, there were of course opportunities to do that, but we never did. No, um, uh, well, macro, and mega plastics ang dami kami
Yes.
nakikita. No, but I'm sure there are microplastics as well, but we never really looked into them.
Okay. Sir, baka gusto niyo pong magbigay ng message. Madami po tayong students sa audience ngayon. Baka pwede po kayo magbigay ng message to inspire the young ones to join the bandwagon and do plankton studies.
Siguro very broad lang siguro, no? Kasi marami na nagsisun bigyan ng message kanina. Actually,
Yes.
siguro in a very broad way, no? We always think that science is, if you want precise, it's objective. Actually, a lot of science is being creative in the way you approach science, no? So there are many things that we don't know yet, no? And there are no fixed ways in addressing your curiosity or questions, no, about nature. There are only ways that are practiced and more practiced and most practiced, no, and it is those people who who dare to create, no, new things that new approaches arise, no. So there are many things that you can do, and don't feel that you are just because we are in a situation now that has to do with health, for example, that only health-related studies are going to be in. No, it's not going to be that way. There will be priorities, of course, but again, you push your imagination to see how you can work on things that you find interesting in the opportunities that are present now. No?
That
Yun lang
is
siguro.
very
Medyo
inspiring,
malalim.
sir. May mga nagte-text po sa akin na nakakaiyak daw po yung message niyo. Okay, so I would like to request everyone to please turn on your cameras. I think we can take a group photo. Yes. Yeah, our IT officer, Sir Paeng, will activate your camera so we can have a photo together. Si Miss Jonaline po si Ma'am Jonaline po yung magpipicture.
Ma'am, pwede pong pa-stop share.
Sorry. Yeah.
Thank you. Just wait lang po for the activation.
Okay.
Meron pa nag-raise ng hand, no?
I mean, nag-raise pa. Sorry po, madami pong questions, sir.
Pwede rin mag-email siguro.
Yes, you can always email Sir Willie for your additional questions. Ay, nasa lab kayo, sir. Hi, lab.
Oo.
Po ba, ma'am, yung pati po ba yung mga attendees?
Yes, please.
We'll just wait po. I cannot see the attendees.
Ma'am, excuse me, good afternoon.
Yes.
Medyo matagal kong i-promote. Isa-isahin natin sila i-promote. Promote pa.
Okay. So matagal. Okay, sige na lang po, never mind. sorry everyone. We can just continue na lang sa program.
Ay, pwede ma'am tayo na lang po, yung panelist na lang po muna.
Sige.
Okay, 1, 2, 3. Isa pa. 1, 2, 3. Okay po.
Okay, thank you. So mag-continue na lang tayo sa program. Okay. So now we will award the certificates to a certificate to our speaker. This certificate is awarded to Dr. Wilfredo L. Campos in recognition of his outstanding contribution as keynote speaker of presentation entitled Using Fish, Egg, and Larval Distributions in Designing Ecological Networks of Marine Protected Areas, or MPAs, at the first lecture series on Philippine Planktology on June 14, 2021, signed by the Director of INFO, the Vice President of PSP, and the Head Administrator of Philippine Planktology page. So thank you, Sir, really, for joining us today and presenting to us a lot of your researches in your career.
Thank you for the opportunity.
Madami pong support from the planktologists around the country. Salamat po, Sir.
Thank you. Thank you.
Okay, so maraming salamat po sa lahat ng nag-join. Again, we are very grateful for your support sa Philippine Planktology page. Kindly wait for the evaluation form that will be posted on our FB page. Maraming salamat din po sa nag-join sa FB Live. Also, don't be sad because we will have our second lecture series soon. We are still thinking of a nice theme, of course, still related to planktology. So please watch out for that. And also, just an announcement, I'd like to tell you that we will be announcing instructions for the membership of the Philippine Plankton Society of the Philippines. So please also watch out for that on our FB page. Maraming salamat po. Thank you so much. Thank you, Sir Willie. Thank you, Director of Info, Sheila Santander de Leon, our co-hosts, Jonalyn Mateo, Dominic Medjudia, Sir Paeng from the ISP, Mark Louis Lopez from the Philippine Planktology page. And of course, Madam Helen Monteclaro, the admin officer of Info. Thank you so much. Hi, Ma'am Bell.
Hi din daw. Hi.
Okay, thank you po.
Okay,
Maraming
bye-bye.
salamat.
Bye.
Bye-bye.
Bye.
John, salamat.
Thank you, Ma'am Chris.
Bye. Salamat.
Thank you po.
Sir Paeng, salamat, Gid.
Thank you, Louie.
Ay, Sir
Thank you
Paeng,
po.
pwede na i-end ang FB Live. Salamat.
Thank you.
Po, bye-bye.
Okay, bye-bye.
Si Algeline, nasa Facebook po yung evaluation. It will be posted later.
Yes po, FB page.